Hello students, today we'll discuss about nucleic acid as genetic information carrier. We'll first discuss the properties of genetic material and a definition of what is a genetic material. Genetic materials are molecules that has the ability to carry information from one generation of an organism to the next. As we know that evolution is possible due to heritable traits and variation, there are four major properties that a genetic material should possess, which are: the molecule should be able to replicate itself, the molecule should be able to store information, the molecule should be able to express the stored information, and the molecule should be able to undergo mutation. Apart from the above four main properties, the genetic material should be chemically and structurally stable. In earlier development towards discovery of the genetic material, scientists believed that it could be DNA, RNA, or maybe protein. In the experiments conducted by Mendel with Pisum sativum, he referred to the genetic materials or genes as factors, as it was not known till then about the chemical nature of the factor that is responsible for transmitting characters from one generation to the next. We will be discussing few of the experiments that were conducted towards finding out the chemical nature of the genetic material. Discovery of transformation. One of the most important and pioneering work was conducted by Griffith on the bacterium Streptococcus pneumoniae in 1928. This bacterium, which causes pneumonia in humans, is normally lethal in mice. However, different strains of this bacterial species have evolved that differ in virulence, that is, in the ability to cause disease or death. In his experiment, Griffith used two strands that are distinguishable by appearance of their colonies when grown in laboratory cultures. In one strand, a normal virulent type, the cells are enclosed in a polysaccharide capsules, giving colonies a smooth appearance. Hence, this strand is labeled as S. In Griffith's other strand, a mutant, non-virulent type that grows in mice but is not lethal, the polysaccharide coat is absent, giving colonies a rough appearance. This strand is called R. Griffith killed some virulent cells by boiling them and injected the heat-killed cells into mice. The mice survived, showing that the carcasses of the cells do not cause death. However, mice injected with a mixture of heat-killed virulent cells and live non-virulent cells did die. Furthermore, live cells could be recovered from the dead mice. These cells gave smooth colonies and were virulent on subsequent injection. Somehow, the cell debris of the boiled S cells had converted the R cells into live S cells. The process is called transformation. However, Griffith did not understand at the time what caused the process of transformation, but named the molecule that is responsible as transforming principle. This same basic technique was then used to determine the nature of the transforming principle, the agent in the cell debris that is specifically responsible for transformation. In another experiment in 1944, Oswald Avery, MacLeod, and McCarthy separated the classes of molecules found in the debris of the dead as cells and tested them for transforming ability one at a time. These tests showed that polysaccharides themselves do not transform the rough cells. Therefore, the polysaccharide coat, although undoubtedly concerned with the pathogenic reaction is only the phenotypic expression of virulence. In screening the different groups, Avery and colleague found that only one class of molecule, that is the DNA, could induce transformation of R cells. They deduced that DNA is the agent that determines the polysaccharide character and hence the pathogenic character. Furthermore, it seemed that providing R cells with S DNA was equivalent to providing these cells with S genes. In the experiment conducted by Avery, MacLeod and McCarthy, they used the virulent Streptococcus pneumoniae 3S virulent strain. They cultured the bacteria in a culture broth and harvested the cells by centrifugation. The harvested cells were then heat-killed and lysed. The lysate was subjected to lipid and carbohydrate extraction. The remaining lysate that contained DNA, RNA, and proteins were tested for the ability to transform the rough strain of Streptococcus pneumoniae into the S strain. They observed that after carbohydrate and lipid extraction, the lysate was still able to transform R into the S strain. They observed that after carbohydrate and lipid extraction, the lysate was still able to transform R into S strain, suggesting that carbohydrates and lipids are not transforming principles or the genetic material. Further, they treated the lysate with Proteus, which digested the proteins that were present in the lysate and tested for its ability to transform the R strain. After removal of proteins, the lysate was still able to transform the R strain into S strain, which indicates that protein is not the transforming principle. This is followed by treatment of the lysate with RNAs that digest RNA present in the lysate. In a similar fashion that was conducted earlier, the lysate was checked for its ability to transform the non-virulent strain to the virulent 3S strain. With observation that the removal of RNA did not deter the lysate of its transforming ability, it was concluded that RNA is also not the genetic material. Finally, after treatment of the lysate with DNAs, the lysate lost its ability to transform, which indicates that DNA is the transforming principle that Griffith coined. The Hershey and Chase experiment. The experiments conducted by Avery and his colleague were definitive, but many scientists were very reluctant to accept DNA rather than protein as the genetic material. The final proof was provided in 1952 by Alfred Hershey and Martha Chase with the use of one-phase virus, which is a T2 bacteriophage. They reasoned that phage infection must involve the introduction or the injection into the bacterium of the specific information that dictates viral reproduction. The phage is relatively simple in molecular constitution. Most of its structure is protein, with DNA contained inside the protein sheet of each head. For the experiment, they labeled the protein part and the DNA with radioactive sulfur and phosphorus respectively. Phosphorus is not found in protein but is an integral part of DNA. Conversely, sulfur is present in proteins but never in DNA. Hershey and Chase incorporated the radioisotopes of phosphorus into phage DNA and that of sulfur into the proteins of a separate phage culture. They then used each phage culture independently to infect E. coli with many virus particles per cell. After sufficient time for injection to take place, they sheared the empty phage carcasses, called the ghost, of the bacterial cells by agitating in a blender. They used centrifugation to separate the bacterial cells from the phage ghosts and then measured the radioactivity in the two fractions. When the radioactive phosphorus labeled phage were used, most of the radioactivity ended up inside the bacterial cell indicating that the phage DNA entered the cells. Radioactive phosphorus can also be recovered from phage progeny. When the labeled sulfur phage were used, most of the radioactive materials ended up in the phage ghost in the supernatant, indicating that the phage protein never entered the bacterial cell. The conclusion is inescapable. DNA is the hereditary material. The phage proteins are mere structural packaging that is discarded after delivering the viral DNA to the bacterial cell. RNA as genetic material. With the help of transformation experiments and bacteriophage infection discussed earlier, it was shown that DNA is the genetic material. However, some viruses do not have DNA as the genetic material but uses RNA as their information carrier. RNA is a nucleic acid that is single-stranded and less stable than the DNA. The first evidence that RNA also has the capacity to carry genetic information came from experiments conducted with tobacco mosaic virus. This virus does not contain any DNA and is composed of RNA surrounded by a hollow cylinder of protein subunits. The experiments conducted by Frankel Conrad in California showing that RNA is the genetic material in this virus will be briefly discussed in this section. In the experiment, tobacco mosaic virus particles were fractionated into RNA and proteins. Later, by using RNA and protein separately in tests for infectivity, it could be shown that RNA alone was able to cause infection. Such a property was not found in protein fraction. This proves that RNA is the genetic material in tobacco mosaic virus. It has been postulated that RNA was the first genetic material formed during the evolution of living organisms. There are certain differences between DNA and RNA that makes DNA a more widely prevalent genetic material as compared to RNA. Difference between DNA and RNA. DNA is a double-stranded molecule that encodes the genetic information used for developing and functioning, whereas RNA is a single-stranded molecule that plays a vital role in coding, decoding, regulation, and expression of genes. Function-wise, DNA is a long-term storage of genetic information, transmission of genetic information to make other cells and new organisms, whereas RNA is used to transfer the genetic code from nucleus to the ribosome to make proteins. It is also used for transmitting genetic information in some organisms and may have been the molecule used to store genetic blueprint in primitive organisms. DNA is composed of deoxyribose sugar, phosphate backbone, adenine, guanine, cytosine, and thymine bases, whereas RNA consists of ribose sugar, phosphate backbone, adenine, guanine, cytosine, and uracil bases. DNA is a double-stranded molecule consisting of a long chain of nucleotides, whereas RNA is usually single-stranded helix consisting of shorter chains of nucleotides. DNA is self-replicating, whereas RNA is synthesized from DNA. In DNA, the bases adenine pairs with thymine, whereas guanine pairs with cytosine. In RNA, the bases adenine pairs with uracil and guanine pairs with cytosine. The deoxyribose sugar and the extra CH bond makes DNA more stable. The small grooves in the double helix model provides minimal place for damaging enzymes to attach. The OH bond in RNA makes the molecule more reactive. It is also not stable under alkaline condition. Also, it has larger grooves that allows enzyme to attach easily. DNA is more susceptible to UV damage, whereas RNA is resistant to UV damage. The helix geometry of DNA is of B form. DNA is completely protected by the body, that is, the body destroys enzymes that cleaves DNA. DNA can be damaged by exposure to ultraviolet rays, whereas the RNA The helix geometry of RNA is of A form. RNA strands are continually made, broken down and reused. RNA is more resistant to damage by ultraviolet rays. In conclusion, DNA, RNA and proteins are three main components that play an important role in living organisms. DNA has been a widely known concept about how it stores our genetic data. However, DNA is not the only component responsible for it. RNA and proteins also play an important role determining the function of a cell. Both DNA and RNA, which are collectively known as nucleic acids, with almost similar chemical constituents, are responsible for carrying genetic information.